Ladies and gentlemen, thank you for joining me again on The Latest With. This is a TV show where we discuss stem cells, and as you can tell, this is a live format. We do take telephone calls from certain individuals, and you can chat online. If you go underneath the camera, underneath the screen, if you look down, it says chat, put in a username, and you can chat and ask questions. So today we're honored to have with us Dr. Aubrey de Grey. I have known a lot about Dr. de Grey's work on an on and off basis. My initial introduction came from Pierluigi Zappacosta. This is a man who founded Logitech and started a company based on life extension. That got me thinking a lot about one of the main reasons why we all do what we do is to make life better. All of the people, all of us in the field. And regardless of religious affiliation or not, we don't want to die. We want to live as healthy, at least me personally, live a healthy life as far as we want. It's a very politically charged issue when you start thinking about these things, but what I've always admired Dr. de Grey from a distance for is because he went out there and he said, look, we live in a society where we understand a lot of things, almost everything. We have almost unlimited resources that we are throwing away at numerous different issues. But when it really comes down to it, we have a shot right now in our generation to significantly extend our lifetime and our life. And Dr. de Grey has put together some very interesting plans, very controversial plans, and which, as viewers here know, we love controversy and we love really interesting subjects. So with that said, please, Dr. de Grey, tell us a little bit about yourself and how you got into this, and then we'll dive into some issues.
Sure. And first of all, I'd like to thank you very much for having me on the show. It's a great honor. I know that you've been doing this for a while and that you've had a lot of very eminent researchers on the show, so I feel honored to be here. Well, I was not originally a biologist. I was originally a computer scientist as an undergraduate, and I switched fields about 15 years ago when I began to discover, first of all, through the woman that I met and married about 20 years ago, and also through many colleagues of hers, I discovered that most biologists actually regard aging as a bit of a boring thing, not very interesting, not very important. I was horrified by this. It was a big surprise to me. And I decided eventually that it was even more important to work in that area than it was to work in the area I was in before, which was artificial intelligence. So I was able to get going and generally influence the field a little bit by offering new explanations for existing data. And then about 10 years ago, I realized a way in which we might go about combating aging that was quite significantly different from the ways that people who were working on the biology of aging had previously considered. Essentially, my proposal was to do periodic repair and maintenance on the body, which is slightly more formally could be described as using regenerative medicine against aging. So restoring the molecular and cellular structure of the body to the state that it was in before it had accumulated the various types of damage that aging consists of. And this is distinct from the normal way that we think about aging, or that people had been thinking about aging, because the more frequent alternative had been to consider the possibility of simply cleaning up metabolism. In other words, slowing down the rate at which these various types of molecular and cellular damage are created in the first place. And people had come to what I think is an extremely correct conclusion, namely that that's actually incredibly difficult to do, because it requires us to understand our metabolism far, far better than we currently do. I realized that even though it sounds as though it ought to be harder, actually reversing the process of accumulating damage may in fact be easier than slowing it down. Because, of course, it's not a case of really reversing the process, it's simply a case of repairing the damage that the process has created, which is not the same thing at all. And over the past 10 years, of course, I have not only fleshed this out, but also talked about it extensively and discussed it a great deal, both within the academic community and in print and in the wider world, and it does seem to be standing the test of time.
Mm-hmm. So tell us, I guess maybe, tell us a little bit more about this. I've read a tad about it. So you believe that there's 7 things we need to do, correct?
Well, let me put it slightly more precisely. I believe that there are many, many, many things we have to do, but I believe that they can usefully be classified into 7 major categories. And of course, there's a billion ways in which you can classify 1,000 things into 7 categories, but the particular classification that I use has a very important feature, namely that within each category, at least I believe, that more or less the same approach can be taken to repairing the particular type of damage. So, for example, one of the categories is cell loss. So cell loss simply means when cells die and they're not automatically replaced by the division and differentiation of other cells. And the solution to that is essentially stem cell therapy. Now, of course, as you and your audience very well know, stem cell therapy is not just one thing. There is a very big field there, and that's because different approaches need to be taken in different tissues to repopulate different organs and different cell types. But yet, stem cell therapy is a field, and the reason it's a field is because different stem cell therapies also have a very great deal in common. So, in other words, it sort of makes sense to treat that as one category of damage within aging. And my big realization about 10 years ago now was that actually all of the various things that go wrong during aging can be classified in this way, and you only come up with basically 7 categories.
So there's 7 categories of things that go on, and then we have different subcategories. And from what I understand, your background is a systems background. You see things like an engineer in some ways. Is that correct?
Well, certainly my background in computer science has influenced the way in which I've addressed the problem of aging, that's for sure. However, I think really the main difference between my approach now and the approach of most people who have spent their entire careers working on the biology of aging is that I'm simply goal-directed. I think of myself as an engineer, yes, but what that means is not so much that I have different skills, it's more that I have different motivations. Most people who work on the biology of aging are doing it as basic scientists. They're interested in finding things out about aging really just for the sake of finding things out, and the possibility of actually being able to use that information for humanitarian good is sort of secondary. It's sort of a bonus.
No, I agree, and that point is actually a very strong point for me. When I first heard about you and your work and I read some of your writings, that really hit a chord because, as some of our viewers know, the reason why I got into science was because my mom, when I was young, had leukemia. And one of the things that really shocked me was learning how 99% of the research that people are running around and doing fundraisers for and running, jogging and doing different things, 99% of that money is going into research that's extremely academic, and then when you have translational research, the majority of it is clinical trials comparing existing drugs at a different concentration. Or does this give you a small advantage to this other drug? And the extreme goal-oriented research, while the public granting agencies are always trying to push it, it is very difficult. And I can talk about it from the field I'm familiar with in immunology and in leukemia. And I agree with your statement at the beginning. My background, when I hear gerontology or when I'm reading my Journal of Immunology and I see a paper on T-cell senescence, I'm like, aging, why do I care? Until I started thinking about and I guess you opened my eyes in many ways because I started thinking about, my God, if we start understanding these processes and classifying them and start dealing with them, we can really do a very big thing. Now, just to cut to the chase, I guess, and I don't want to be rude on the program, but tell me a little bit practically, what exactly is, I mean, I've read some of your papers, and I've also read, I think, an MIT paper where they criticize you. And I'm thinking, why would somebody criticize? Your point was a very straightforward point, which, how can somebody criticize it? There's 7 aspects. What is somebody going to argue that mitochondrial damage doesn't occur in aging, or that we shouldn't be trying to fix extracellular aggregates? But my question to you is, how, and I should have done more homework, I didn't before the show, but how are you going about doing this, and how can we help you as a scientific organization?
Sure. Okay, so a couple of things. First of all, I do want to make clear that I don't in any way think that basic curiosity-driven research is overdone. I think that the work that still needs to be done to understand aging better is very substantial indeed. So I'm very pleased that there are lots of people working on that. It's just that there ought also to be lots of people working on the translational research to actually apply that knowledge to produce new therapies. And a lot of what's required there is not really so much throwing new money at these problems. It's really a case of repurposing the work, the expertise that's already in place and that's being developed in a lot of other areas of medicine and of biology, and just educating helping the researchers involved to understand that their expertise is more applicable to the general problem of aging than they might originally have thought it was.
Absolutely, I agree 100%.
Okay. In terms of the criticisms that have been raised, so you mentioned that there was a critical article in MIT Technology Review. That's certainly true. A couple of years ago, about 5 years ago now, there were a couple of rather strident critiques of my work. It wasn't really a problem of a disagreement about what the nature of aging was. Certainly there was an acceptance that, for example, mitochondrial mutations accumulate and they're bad for you, and extracellular aggregates accumulate and they're bad for you, and so on. The problem was skepticism with regard to the particular solutions that I was proposing. Mm-hmm. And by and large, this skepticism arose from the critics being simply ignorant about what had already been done. Because, as I just said a moment ago, a lot of the work that's been done that's of relevance to all of this, and from which we need to start in order to develop the therapies that I proposed, has been done for other reasons. It's not been done by people who study the biology of aging, mm-hmm. or who would think of themselves as card-carrying gerontologists, and therefore it's been done by people whose papers just don't really fall into the hands of gerontologists very much. Now, I could be uncharitable and say, well, my critics should actually have, as such, read my own papers a little bit, and perhaps even read, dare I say it, the references and the experimental work that I cited in those papers before coming down so hard on me. But, well, they know better now. They don't do it anymore. So I guess I've sort of won that battle. But yeah, I mean, of course, the details of what I proposed are very extensive. I don't know if you've already mentioned that I had a book out a few years ago, Ending Aging, which covers all of this, and it covers it in a great deal of detail. Each of the 7 different categories of damage that I describe in SENS is addressed in a whole chapter of that book, and of course the ways in which we propose to repair and or obviate that particular type of damage. So yeah, that's really the key, that there's so much detail, that we already have so much precursor technology, if you like, so much background technology from which to start in order to develop therapies that really ought to work to eliminate these various types of damage.
No, I think, okay, so let's dive in. I mean, you've done a very, very cool thing, which is this, how do you pronounce the Methuselah prize?
Yeah, Methuselah mouse prize.
Yes, and so these strategies, you've applied them to the mouse. How long, what kind of life difference do we see?
Okay, so I should be rather clear here. The Methuselah mouse prize was never really an attempt to get the 7 SENS strands developed. Mm-hmm. The Methuselah mouse prize was put forward, was something that was created and administered by the Methuselah Foundation starting in 2003. The Methuselah Foundation was a charity started by myself and a businessman named Dave Goebel. And it was really a PR exercise, an attempt to raise the profile of longevity research in the eyes of the general public, in the eyes of people who really are, turned off by science, by the hardcore aspect of science, but they're very much more identified with things like world records and prizes and so on. And the research that needs to be done to actually implement SENS, implement the 7 types of repair that I talk about, is something that is not going to win the Methuselah Mouse Prize anytime soon, probably not for several years. The Methuselah Mouse Prize was specifically constructed so as to be winnable by incremental improvements that might be achieved using the much more straightforward techniques that researchers in the biology of aging are already exploring, but which will not scale and will not actually give appreciable benefit to humans in the future. Mm-hmm. So there's a very big separation there. In fact, that separation ended up being quite a source of tension within the Methuselah Foundation, because when we started not just doing these prizes, but also being a regular funding body and actually putting money into specific research projects, the way in which we needed to talk about the foundation's mission and generally do outreach and messaging and so on was very different for the two activities. For the research activities, obviously, we had to make sure that everybody knew that we were really responsible and knowledgeable and knew what sorts of things to put money into, whereas for the prizes, the priority was to be much more, populist and glitzy. And those two things really didn't go together very well. So that was actually the main reason why in 2009 we created a new foundation called SENS Foundation, which, and the Methuselah Foundation divested all of its research activities into the new foundation. So the Methuselah Foundation very much still exists, and it's very effectively pursuing the prize alternative, which I very much still believe in. But this new foundation's been created, SENS Foundation, which is doing all the research side. And it's working really well in terms of messaging and so on. The fact that we can now be independent organizations, independently appealing to different sources of motivation, if you like.
Now, we have a lot of our viewers are biotech people, and we don't want to give the impression that what we're talking about here is some speculation and some theoretical matters. This anti-aging is big, big money. The company Sirtris was bought by what? By GSK, and it had a phase 1 clinical program, was bought by, I think, $1 billion+, or 750 million, despite being in phase 1, with the resveratrol through a different delivery method, because they had the intellectual property on the sirtuin, I believe, which causes life extension in the fruit fly. Do you remember, Aubrey, how long does the life extension, and do you know anything about that story? It's a little bit vague in my mind. I met some of the principals that I think it was, 2 years it took them. They in-licensed the IP, formulated it, got some really cool preclinical data. They were looking at, I think, type 2 diabetes was their indication, and boom, 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 sold out. A biotech dream for many people. Tell us a little bit about that story, or your take on the story.
Well, yeah, I mean, of course, I'm very familiar with all of this, and I think that the work that Sirtris have been doing and the work that David Sinclair does in his lab at Harvard is extremely valuable work, no question. I don't think that it's likely to result in drugs that can substantially extend the human lifespan for most people. But if it can do what it has already, if that general approach can do what it already looks as though it probably can do, which is to constitute a pretty good treatment for type 2 diabetes, then, more power to them as far as I'm concerned. The only difficulty, of course, is if people overinterpret the generality of this work and they think, well, this has been solved for a lot of money, therefore aging has been cured, therefore we don't need to do anything else anymore.
Of course.
But most people don't seem to be making that mistake. So, I feel generally positive about Sirtris. Now,
one of your out of those 7 causes, one of them is I saw age, advanced glycation.
Advanced glycation, yes.
Okay, what's the story with those ones?
All right, so advanced glycation end products are chemical linkages between proteins that are created spontaneously without the help of enzymes as a result of spontaneous chemical reactions between proteins and sugars. And the reason that they're interesting in aging is because they occur quite a lot in the extracellular matrix. Which consists of proteins that are laid down early in life and, for the most part, are not recycled throughout life. So they just sit there, and they get more and more cross-linked. And the reason this is a problem in aging is because, in many cases, the extracellular matrix needs to be elastic in order to do its job. So if we think of the walls of our major arteries, for example, they need to be elastic in order to respond to the constant pulsation of the heartbeat. And that elasticity gradually diminishes as a result of the accumulation of these crosslinks. Basically, they make the artery wall stiffer. Exactly the same process goes on in the eye and makes the lens of the eye stiffer. And that's the reason why we end up not being able to see things close up when we get older. So then the question is, what do we do about this? The reason these things are called advanced glycation end products is because glycation is the word that was coined about 30 years ago now to describe this process of what in more polysyllabic terms is called non-enzymatic glycosylation, the reaction of sugars with proteins, but without enzymes. We're interested in doing something about that. We're interested in breaking those crosslinks. And the good news is that it ought to be possible to identify drugs that can do this, because the chemical structure of these crosslinks is very unusual. It's very different from anything that we see in that the body lays down on purpose, so to speak. Which means that we've got a respectable chance of being able to actually identify drugs that are sufficiently selective that they won't be toxic.
Yes, last time, very quickly, last time on the show we had Larry Goldstein, and he made a lot of money, not in stem cells, what he's known a lot for, but, well, I don't know if he made a lot of money, but had a very nice company where they set up in vitro system of screening drugs for ability to modulate kinesins. Do they have, I mean, how I see your book in many ways is that it's like a Bible of all sorts of really neat research directions. Let me ask this question. Can you do in vitro screening for inhibiting the age and interaction with the RAGE receptor?
Certainly we can do in vitro screening for what we're looking for. However, what we're looking for is not interaction with the receptor for AGEs, the RAGE receptor as it's called, or indeed the inhibition of the creation of these crosslinks. What we're looking for is something that can break these crosslinks after they've been created.
I see.
It's probably going to be relatively easy to identify drugs that can slow down the creation of these crosslinks, and indeed there are a number of drugs around already that appear to have that property. But we're interested in taking people who have the misfortune to be already in middle age or older, and therefore who have accumulated a lot of these crosslinks already, and we'd like to rejuvenate them.
I see. So, but just to go back, I guess, to general a little bit, with your thinking and your work and what you've been doing, you've identified how many other factors besides age? Age is an example. How many other things in general that are tangible things that our viewership can start thinking about?
So as I told you, there are 7 major categories of damage.
Yeah.
So AGEs, these advanced glycation end products, turn out to be responsible for the great majority of the cross-linking that accumulates during aging. There are other mechanisms that can form cross-links in the extracellular matrix that accumulate during aging, but they account for only a very small proportion of the process. So we know what our targets are. They are basically, in fact, there's just one AGE, something called glucosapine, that is very definitely our major target because it is by far the most abundant of these crosslinks. And in general, that's the sort of rule. If you look at the other types of damage, for example, accumulation of molecular garbage inside the cell, in other words, byproducts of normal metabolic processes that the cell makes but then does not know how to destroy or to excrete. This is basically the reason why we get cardiovascular disease. because there are various oxidation derivatives of cholesterol that macrophages don't know how to process properly. And there are lots and lots of different ones, lots and lots of different oxidized cholesterol derivatives, but there's one called 7-ketocholesterol, which appears to be by far the most important, the public enemy number one, because it's more abundant and it's more toxic than the others. And so in our research center in Mountain View, we are conducting quite a lot of work, and also in a lab that we fund extramurally at Rice University in Houston, looking specifically at identifying non-human enzymes that can break down 7-ketocholesterol. And we're not really bothered about the minor players that are also accumulating but much more slowly.
I see. So, okay, so you inhibit intracellular accumulation of this, but I don't understand.
Again, not inhibit, repair. We reverse the accumulation. We take cells that have already accumulated stuff and we get rid of it.
But how would you practically deliver this enzyme in the cell?
Well, that depends on the tissue. So, so the example I'm talking about at the moment is in the case of cardiovascular disease. Now, in this case, I expect that the most straightforward way to deliver it will be through genetic modification of hematopoietic stem cells, the blood stem cells, because the cells that we need to modify, that we need to confer on them the property of being able to destroy this stuff, those cells are macrophages. Of course, macrophages are a type of white blood cell. So if we can simply do a bone marrow transplant with autologous, of course, cells taken from the patient that have been modified ex vivo to incorporate an additional gene or two encoding the enzymes that can break down these nasty cholesterol derivatives, then we reintroduce those stem cells back into the same person. Then after that, all of their blood, including all of their macrophages, will express these enzymes, and so we'll have an augmented catabolic arsenal, and we'll be able to get rid of this stuff. And existing atherosclerotic plaques, even atherosclerotic plaques that are quite advanced and have a big acellular necrotic core, will be predicted to shrink, because macrophages will be burrowing into the atherosclerotic plaque, same as they do at the moment. But rather than simply becoming part of the problem, because they can't break down that material any more than the previous macrophages could, these new macrophages that have been modified in this genetic manner will be able to gobble things up, and they may, and they will thereby gradually shrink the plaque. That's the idea. That's the whole.
This is now you are talking the language that all of us here really love to hear. The bone marrow transplant and the stem cell. Let me ask you, because it's very exciting what you're talking about. How do the stems, first question in my mind, has anybody done this experiment? Before we got the microphone working, I was doing my little monologue here, ranting about stem cells. Has anybody done the experiment where you take an old mouse, and syngeneic mouse, genetically the same, and you give to the old mouse hematopoietic stem cells from a young mouse? Does that cause any life extension?
Okay, so yeah, experiments along those lines have been done. There is a problem, which is that in a normal mouse, the stem cells, even if they're not quite as healthy as they used to be, have a tendency to want to stay put in the stem cell niche in the bone marrow.
Yeah.
So if you put a bunch of if you just inject a bunch of stem cells into an old mouse, they basically don't engraft, and they just go away. Now, I know that one of the things you mentioned before I was able to get my sound working was some work that you mentioned in Irv Weissman's lab. There was some other work in Irv Weissman's lab not long ago, which was actually conceived by a guy named Daniel Kraft, who is one of our research advisors. And that involved using antibodies to essentially deplete the hematopoietic stem cell niche.
Yes.
Antibodies that reacted with a particular cell surface marker of blood stem cells, and did so in a manner that caused them to die, basically. So you ended up with a situation in which if you co-administer new stem cells, then the new stem cells are able to engraft because the niches into which they would like to engraft are actually vacant.
Yeah.
And it's taken a bit of time to get this really working properly. Initially, it could only be done in skid mice with no immune system. And it's taken a while to refine it so that it works in immunocompetent mice. But I believe that there's good progress in that area now. And certainly what I would like to see, is, for a whole bunch of reasons, is the ability to repopulate the stem cell niches of not just the blood, but also other rapidly renewing tissues, such as the gut and the skin. And we are involved in some projects in all of those areas.
Wow, this is very, very interesting. But this is extremely interesting. Let's talk about the other stem cells. So when you think about, first question, we always talk about Hayflick limit. In an old person, what percentage of the cells have even reached the Hayflick limit? I'm just curious. And somebody who's 80 years old.
Yeah, my belief is that the number is very, very small. Virtually all cells that divide a lot, either they've become cancerous and therefore they don't have a Hayflick limit anymore because they've turned telomerase on, or they die because they apoptose because of the systems that exist. Only a rather small number of them get to undergo what we think of as replicative senescence of the sort that we see in vitro. And in fact, even the cells that, from a phenotypic point of view, look as though they might have actually reached replicative senescence, it's often quite hard to tell whether they have, because, as you probably know, cells can get into that state simply as a result of DNA damage, double-strand breaks especially, even if they haven't divided very often. And my personal belief is that the overwhelming majority of cells that are seen to be exhibiting some of the gene expression patterns characteristic of replicative senescence have actually got that way through DNA damage rather than through dividing a lot.
Okay, so in these,
ultimately what we have is the situation where the cells that are used, that are famous for exhibiting replicative senescence in vitro, are cells that simply don't need to divide very often in vivo. Typically dermal fibroblasts. The epidermis obviously is a constantly renewing tissue, but the dermis, the lower layer of the skin, is not. You only have cell division there at an appreciable rate on demand when, for example, there's a wound, as opposed to anything that's actually going on constitutively. So it's sort of reasonable that fibroblasts would not have the capacity to overexpress telomerase when they were, if you like, tricked into thinking that they were in an infinite-sized wound, which is what cell culture functionally is.
Wow, that's a very cool cell culture functional analysis of fibroblasts in infinitely sized wound.
That's what I think of cell culture. Yes, I mean, think of yourself as a fibroblast. When do you divide? You divide when you want to close a wound, and contact inhibition constitutes the process, which obviously is when cells stop dividing in vitro, is exactly the same as closing a wound. If you want the cells to divide indefinitely, you just dilute them so that they never reach contact inhibition, which is just like being in an infinite-sized wound.
Interesting. Alexei here, who is a hematologist, stem cell scientist, asks the question, has anybody done the relationship between stem cells and aging? The function of stem cells, no, not aging, longevity. People who have a more potent stem cell compartment live longer. Do you know anything about that?
Well, there's certainly quite a lot of work being done in mice in that area. There are, of course, lots of different inbred strains of mice which have a whole bunch of different genetic characteristics. And, of course, one of the characteristics that differs between different strains is longevity. I think the most pioneering work in this area was done in Kentucky by Gary Van Zandt, but there's been quite a lot of work since that time. That was about 20 years ago when he started in this area. But essentially, it was shown that, yes, absolutely, there is a change in stem cell number and stem cell function during aging. And the change is not really what you would expect. In most mouse strains, actually, the number of long-term repopulating cells in the bone marrow goes up during aging. You actually get more and more of them as time goes on. And there are all manner of different hypotheses out there for why this might happen. Nobody really knows, as far as I'm aware.
But I see. The number of colony-forming units goes up in an older mouse.
That's right.
Really? I didn't know that.
In most strains, yes.
Which mouse is winning the Methuselah prize right now anyway?
Well, okay, there were two Methuselah mouse prizes. The first one that we set up was just a very simple headline thing that said, you've got one mouse, the individual mouse world record. Mm-hmm. And that is held by a mouse that lived very nearly 5 years, 1 week shy of 5 years. Uh-huh. The mouse in question was a growth hormone receptor knockout mm-hmm. created in the laboratory of Andrew Barkey in Illinois. And it was absolutely tiny. These mice are always very small, but this one was particularly small. And it did appear to live almost 5 years. Now, that is so far off the scale. It's probably 6 months longer than any other mouse, mus musculus, of course.
What would that translate into in human years?
I'd say 180, something like
180.
That. So it's so far off the scale. It's probably 6 months longer than any other mouse has been verified to live. That if it hadn't been reported by one of the most reputable and most reliable researchers in the whole field, then people would just have not believed that it was true. But the fact is, it was reported by such a person, so most people believe it. However, what's more interesting is the other prize. We created a second prize because we wanted one that was more scientifically robust, if you like, and mm-hmm. also more focused on our own goals. So the second prize was not for just one mouse, it was for a cohort. Basically, you wanted to show that the thing was reproducible. So we used the conventional gerontological definition of maximum lifespan, which is the average lifespan of the longest living 10% of a cohort in an experiment. Uh-huh. We said that's what you need to beat the record of. And we also said that the experiment had to be published in a proper peer-reviewed journal, so it wasn't just one mouse we were talking about anymore. But the final thing that we threw in as a criterion for this particular prize was that the intervention that was done on these mice had to begin only in the second half of life. Okay. So basically that meant starting 18 months of age or after. Because we were interested in things that could be applied to individuals that are already getting on. And the current record holder there is from an experiment that was published in about 2003 by Steve Spindler at University of California, Riverside. Spindler did a very careful experiment of calorie restriction starting at 19 months of age. Now, calorie restriction experiments have, of course, been done innumerably over the years, ever since the 1930s, mm-hmm. but quite a lot has been discovered over the past few decades about how to keep mice happy, how to introduce calorie restriction in a minimally stressful way, for example. Mm-hmm. And experiments starting late in life had not been tried for a long time. So Steve basically threw all the modern knowledge and expertise at this problem and tried it, and he got a very respectable longevity increase of about 4 months compared to the ad libitum fed animals. Yeah, that was the record holder. That still is the record holder for the second prize.
So in these kind of interventions, do you think there's an increased stem cell compartment?
That, as far as I know, has actually not been explored. I don't know whether calorie restriction causes an increase in the number of long-term repopulating cells in the bone marrow. That would be a lovely thing to ask, and I don't know whether anyone's ever asked that question.
We have a question here about insulin being the key protein by which caloric restriction works. Is that true? Insulin receptor signaling?
I think it's safe to say that nobody has the faintest idea how calorie restriction works. And that's sort of how it ought to be, because calorie restriction is an evolved response. The purpose of calorie restriction is, we have a lot of machinery out there that causes the body to be able to respond to nutrient deprivation in a manner that minimizes the diminution of fitness that would result. Stops starvation, stops death of any sort. So we would expect the situation that we see. In other words, that there are a few, if you like, master control genes that are involved in the initial sensing of the nutrient deprivation situation, but that those genes are at the pinnacle of a big, cascade of downstream effector pathways that actually cause aging to be slowed down.
Mm-hmm.
And those downstream effector pathways are very, very, very poorly understood still. There's an enormous amount still to be discovered in that area. We understand the master control genes like the sirtuins and the insulin IGF pathway, reasonably well at this point, but what happens after that is still a big, big black box.
No,
I would say the best way to look at calorie restriction is that it's a very multifarious phenomenon in which an awful lot of processes are coordinately modified, just in the same way that aging itself is a very multifarious phenomenon.
No, the caloric restriction is very interesting. I was trying to put together a paper one time on caloric restriction, and I was trying to find out something wrong with it. And it's very interesting. It increases angiogenesis. Via adiponectin, it actually inhibits depression. There's such a wide variety of interesting benefits. Let's see here. There's a couple more quick comments. Boris, people extend mouse life by superexpression of tumor suppressor genes, such as p53, or giving rapamycin. Yeah, there was a paper. Wasn't there a paper where Telomerase in the Journal of Clinical Investigation? What was the genes? They had a really cool gene combo there.
Right. Okay. So yes, another of the advisors on our research advisory board, Maria Blasco, and her husband and collaborator Manuel Serrano did an extraordinarily valuable study a few years ago, which you're referring to. They were interested in the role of telomerase in protecting mice from aging in the context where they were not getting cancer. So, of course, normal lab mice mostly die of cancer.
Right. And
that is a massive confounder, a massive problem for actually studying things that don't have to do with cancer in mice. But what Blasco and Serrano were able to do was create a mouse which basically never gets cancer. They arranged for it to overexpress both p53 and also p16, p16F, which are two arms of the tumor suppression machinery, and the suppression of cancer was pretty much total. So what they did, having done that, was they then threw in telomerase. They threw in an extra copy of telomerase. As you say, they did get a very significant increase in longevity of these mice. Now, I definitely think there's a case for doing a lot of repetition of that experiment, in particular doing it in other strains, longer-lived strains, and so on. But as things stand, I think it's a real milestone experiment, because it's a really classic case where it was rationally designed, if you like, where people said, okay, there are these multiple things going wrong, we need to fix them all, so let's do this multiple intervention, and it actually worked.
Yeah, no, exactly. Really quickly, so all these gene interventions, how far are we from giving telomerase to people? Can we give telomerase to people, or it just causes cancers? Silly question, I'm sure, but...
Well, it's not a silly question. No, it's a very topical question indeed. Of course, at the moment, we don't have safe and effective somatic gene therapy, so giving the gene for telomerase to people is not really something that we can contemplate right now. But there is the alternative, since obviously we already have the gene, of giving people drugs that stimulate the expression of telomerase. And that is a process that absolutely people have been looking at quite a lot over the past several years. There are a number of companies out there now. So Sierra Sciences, which you mentioned in your introduction, the company funded by Pierluigi Zappacosta, has been looking at this for the best part of a decade. Another company named TA Sciences has a compound now, TA65, which is an extract from an oriental herb that appears to have a significant effect on the expression of telomerase. And, at the moment, I've got to say the jury is out. We do have significant reasons to believe, both from in vitro work and from actual in vivo work with TA65, that the immune system can be significantly rejuvenated by stimulating telomerase expression. Whether that's a good thing or not, over the long term, of course, can't be determined until we've done really long-term studies, which certainly have not yet been done. And there is always the risk in everyone's mind that over the long term, the constitutive overexpression of telomerase, or the indiscriminate overexpression, shall we say, may be permissive to cancer. But certainly, as things stand, there is no firm evidence that that's actually occurring. And actually, the people who are marketing TA65 are making great play of the fact that that it looks, from the, albeit decidedly anecdotal data that they have so far, as if cancer incidence may actually be reduced in the people that are getting this drug. This is
a very, very interesting point. As you may know, our company, MediStem Inc., we have stem cells from the menstrual blood that we're developing. Yeah. High proliferating tissue, very angiogenic, a lot of blood vessels. We're going to the FDA to ask permission to use the cells in patients whose legs will be amputated, who have bad circulation. And obviously, as CEO of the company, my biggest concern is you have these highly angiogenic cells. Biggest concern in our development is, well, what if they activate dormant tumors? In our initial series of studies in a spontaneous skin cancer, administration of these cells decreased the spontaneous incidence of cancer. And then we did a subsequent study in glioma, which we published. It was a more detailed investigation, and we actually found inhibition of the glioma growth, both by intratumoral and intravenous administration. So like you said, the question of cancer and its stimulation is a very bizarre one, because these data were completely counterintuitive. But okay, we're going to have to wrap up pretty soon. I'm sure you may be getting tired, but a couple really quick points. Right now, there's stem cell therapy being used clinically, experimentally, of course, under clinical trials. What do you think about the importance of circulating endothelial progenitor cells? There was studies by this guy, Jay Edelberg, where he did some really neat work giving, I believe it was 8-week-old, anyway, let's just say young bone marrow cells as a source of endothelial progenitor cells, giving them to old mice. Now, the old mice, when you would do a heart transplant, putting a young heart in the old mouse, it wouldn't integrate properly. When you would put the old bone marrow, it still wouldn't integrate. When you put a young bone marrow, the bone marrow would somehow work with the old body and integrate the young heart into it. What do you think about endothelial progenitor cells as a mechanism of reversing the aging process, at least vascular aging?
Right. So, I mean, obviously, I don't want to speculate too much because, as you've already indicated, it's still very early days in the exploration of endothelial progenitor cells. Mm-hmm. But I'm pretty damned excited, to be perfectly honest. It's not just Edelberg's work that you just mentioned. There's a bunch of people working with endothelial progenitor cells and coming up with results that I think are very promising indeed, as ways of repopulating things. I think they could be they could be as powerful as mesenchymal stem cells are being found to be, but in different ways. So I think they could be a really important part of the stem cell therapy arsenal quite soon.
You know, one thing that might actually be fun, one of the advantages of having a show like this is that we are live and we have access to a lot of people. So if we have 2 minutes, let's call Amit Patel up. We'll see if he answers the phone. I don't know if he's there, but let's ask him, because Amit's been injecting stem cells. I believe in over 1,000 people he injected stem cells. And some of our other friends who have injected stem cells have reported, I can say this non-confidentially, there have been some reports of people receiving stem cell therapy and having other benefits, including arguably the gray hair reversing. Have you ever heard of that, by the way?
Well, I haven't heard of that, no, but an awful lot of rejuvenative effects have been reported anecdotally by 1 or 2 people as a result of stem cell treatment. And even if the stem cell treatment in general doesn't work, it doesn't surprise me very much that there could be occasional cases of benefits that cannot be reproduced.
Let's see really quickly if Amit's here, because him and I were talking, and about these issues. He might be in surgery. He's a cardiothoracic surgeon in Utah. And it's 6 o'clock now. We'll see if he's here. I don't know if you can hear the phone ringing on the. But I just wanted to ask his opinion on endothelial progenitor cells for vascular aging. Oh, he's not here. We'll leave a message off. Let's just see if he's here. Hi, Amit. It's Tom and Aubrey DeGray, and the whole world is watching me leave this message. We're just wondering about endothelial progenitor cells and aging. Give me a call back on my cell phone if you are not in surgery. Okay, bye, Amit. Okay, here's very quickly the ideas that I wanted to bounce off you. Is I have to read your book, and I started reading that paper that you sent me. Through the viewership of this show and a lot of the relations that our viewers have made, we know there's a lot of very wealthy people interested in this field of regeneration and increasing the human lifespan. Through preliminary discussions with them, the biggest obstacle that I found has been that they say there is no credible roadmap. They say, we would love to put money, we don't want to die. We don't. A lot of the people say, look, we believe in God, but we're not going to go to heaven. That's supposed to be a joke. No, but seriously, a lot of people say we have, as a humanity, so much funds. How can we advance this field? Who are the players? What are the organizations? Maybe you can even tell us limiting factors, because from what I know of you and your book, it seems like there is a roadmap. You know, people cannot say that it's not peer-reviewed. You have 102 publications on PubMed, which is 27 more than I have. But but tell us, what can we do to help? I mean, for the next step.
Well, you're absolutely right. I mean, certainly SENS is a roadmap. It is absolutely, explicitly a roadmap.
What is SENS for our viewers who don't know that?
Okay, so SENS is an acronym. It stands for Strategies for Engineered Negligible Senescence. So that probably tells you why I don't often spell it out. But yes, the proposal I've put forward, this 7-point plan for repairing the molecular and cellular damage of aging, is very much a roadmap. And there are bits of it that doubtless will end up hitting obstacles that we hadn't anticipated. This is what happens when you develop any pioneering technology. But in my view, it is a sufficiently detailed roadmap as things stand that it absolutely merits extensive funding, extensive exploration over with a great deal of urgency. Mm-hmm. And of course, I spend a lot of my time trying to persuade people of this and getting them to pursue it. Some of the problems that I alluded to earlier to do with the different mindsets of curiosity-driven scientists versus goal-directed technologists have contributed to the difficulties in communicating the credibility of SENS, but that problem, as I mentioned, is pretty much over. So I'm pretty optimistic that over the next few years we are going to see a real landslide of funding, especially from philanthropic sources, but also from the private sector and maybe even from government, in actually applying regenerative medicine effectively and comprehensively to the problems of aging.
So we can get involved in SENS then, or what? I mean, I'm just thinking for our viewers and for myself personally. Next step after this conversation.
Next step is go to our website. Go to our website, read my book. You know, you can find out all about it that way. Really, you know, it's. The information is out there. We've been at this. I've been at this for 10 years now or more. You know, getting that information set out in both academic and more general audience terms, and there's no excuse for not knowing about it anymore.
We have no excuse. There are some of the books here from Amazon about this that popped up on my screen here. Ending Aging. Oh, the audience can see it on the screen. the Immortality Institute, the Scientific Conquest of Death, Countdown to Immortality. Very, very interesting. And I'll tell you, Aubrey, you know, a lot of the people who watch this show are very advanced in the area of stem cell and regeneration, and at a conceptual level are very big supporters of everything that you're talking about. But yet, a lot of times when I said, you know, I have Aubrey de Grey coming on, a lot of them said, who's Aubrey de Grey? So we need to figure out some way of communicating with everybody. And, you know, if you'd be interested, we love to write numerous papers. Maybe we should co-author a paper together, at least trying to put some of what we know and some of what you know, and trying to figure out. The one last thing that was really interesting, it popped up here on the chat, is from somebody who said, I had stem cell therapy for multiple sclerosis, and the injected cells made my nails and hair grow faster. Crazy stuff. It's really something that I would love, you know, Aubrey, I would love in the future, us to continue a dialogue, us to start feeding you some of the things that we've seen and our colleagues have seen, and figuring out some ways of, I mean, why I'm saying this is because there are numerous trophic activities that happen when you inject stem cells clinically. And even at University of Utah, one of Amit Patel's colleagues was telling me that they're injecting stem cells in the heart intracardially, but they're seeing effects on the type 2 diabetes. So there's a lot of very interesting rejuvenation things, but in my mind, I still think we need to understand all this extracellular stuff that's going on, because I'm sure, like you mentioned, there's enzymes or there's different adjuvants that we can inject to make our therapies work substantially better. You know, I mean, one quick example is when you do the bone marrow transplant, and you do male bone marrow into the female, and then when the female dies of of something else, and you start looking, you see those the male chromosome expressing cells all over in the female's body. So there is an extent of rejuvenation, but the rejuvenation, I don't think we've optimized it nearly as much. Now, the last thing before we go, and now I'm going to say something really crazy, but I love, here is the SENS website that's popping online. Raj put it up there. But yeah, we'll put on our website, we'll put the link to the SENS Foundation. You have Tony Atala as a research advisor on this?
Oh, yeah.
Oh, wow. This is excellent. We definitely, yeah, Maria Blasco, we definitely have to work on getting this this out. You know, Aubrey, another thing that would be really fun to do is getting some of your other collaborators up and having discussions on our website here, on our TV show. But last thing before we go, and this is kind of crazy, but I never thought I'd speak with you in such a format, so I figured I'd ask you. In my mind, Alexis Carrel, from what I understood, was able to extend the chicken heart's life quite substantially by keeping it perfused ex vivo. First of all, is that a correct statement?
I doubt it, personally. I think that the evidence at this point points to the much most likely explanation being that new cells were introduced into that heart periodically, some perhaps by accident or perhaps by technicians who just wanted to keep Carrel happy. But I don't think that experiment could be reproduced rigorously now. That's even more fascinating in and of itself. If new cells were introduced, I mean, somehow they must have been integrating and supporting the function, no? I agree, but this is chickens, remember. This is different from the way that the heart works, a bit different from the way that the heart works in a human being.
You know, with Alexis Carrel, just a quick note to our viewers, Alexis Carrel is the only Nobel Prize winner who actually documented a religious miracle. He was a very astute Catholic, and the irony of all of this is in his publications in Journal of Experimental Medicine, like 1912, he's talking about embryonic stem cells. So it's a very interesting aside, but my question to you was this, Aubrey. It seems to be more easier to keep just the brain alive outside of the body than to try to keep the whole body immortal.
I don't think so. I don't think it's going to be all that much easier. I think the brain is the most complicated organ to keep alive, actually, because you have to not only worry about the cells themselves, but also these very sophisticated interconnections between the cells. And the things that go wrong in the brain are very multifarious. Of course, you have molecular garbage accumulating inside neurons, also outside neurons, such as senile plaques. You also have loss of neurons and loss of synaptic connections. I think the brain's pretty tricky. And I think by the time we get to be able to keep the brain going really well, we will have solved pretty much everything else.
I see. Okay. Yeah, no, it's just something I was picturing Alexis's heart beating and then keeping the brain. Raj, our producer, is showing on the television screen that Aubrey Dugré gave a keynote talk at TED, at the TED conference. There's a picture of Aubrey right now speaking. This is definitely something, Aubrey, if you'd like to keep in touch with us, we would love to. So before we wrap up, let's ask the audience if there's any last questions. I'm watching the audience. And Aubrey, is there anything else you'd like to say before we get going?
I don't think so. I think I'm pretty much done. Thank you very much for your time.
Thank you so much for staying awake this late to chat with us. And keep us apprised of your schedule. You said you come to San Francisco quite regularly. We'll fly up and meet you. Dr. Dugré, thank you so much.